Hi, my name is Aarohi Usapkar, and I'm a senior at Doherty Valley High School in California. I did my project on identifying the genetic correlates of colorectal cancer, and I worked with Connor Dunn as my mentor. To start off, I want to introduce the topic that I was trying to research. Colorectal cancer is the third most common cancer worldwide, and it's caused by cell overgrowth in the colon, rectum, or large intestine. Often, overgrowth can be seen as precancerous polyps months or even years before actual cancerous growth ever occurs. And this can happen because of lifestyle and or acquired and inherited mutations, which can affect patient survival in various different ways. This was the primary focus of my research. I wanted to try and explore how genetic mutations that people have been knowing have occurred, how that could possibly affect patient survival quantitatively. So mutations that I found could be in oncogenes or tumor suppressor genes. And once they're mutated, the way the genes are used can vary in multiple different ways. So, my project was based off of statistical analysis. I used existing data from a lot of other cancer databases to collect data on different genes and try to estimate gene relevance using Kaplan-Meier curves and log-rank tests. So I used cBioPortal and I collected data from the Memorial Sloan Kettering database. And I used graph comparisons and literature to estimate which genes I could possibly find relevant. So using existing research methods, I was able to narrow down to four genes, which were APC, KRAS, BRAF, and RNF43. And then I used Kaplan-Meier survival analysis and a log-rank test to calculate the p-value. And depending on that, that's how I estimated whether or not a gene was relevant. So the first gene that I explored was APC. APC is a tumor suppressor gene that's associated with the Wnt signaling pathway. So basically, its purpose is to decrease excessive cell growth. So when it's mutated, it can lead to the accumulation of B-catenin and the growth of adenomas, which are cellular growths that can lead to cancer. So mutation types and counts can vary. There can be multiple APC mutations within one cell, and there can also be multiple types depending on where the mutation occurs. The most often mutation I found was truncating, um, and these are associated with many other syndromes. A lot of these syndromes can be familial, so they're genetic and they're passed down. And some of them can be affected depending on the location, so they can occur in the colon, the rectum, or the large intestine. Often all three are usually linked, but for this mutation, because we can't narrow down where it specifically is located, sometimes it's difficult to tell. What I was able to get from the data was the average survival times. And for wild type patients, which is patients with no mutation, it was 32.95 months. And for mutated patients, survival time was 41.34 months, which indicates that having an APC mutation actually does improve survival. This was slightly controversial because a lot of research has conflicting opinions on this, depending on what kind of mutation the APC was. Sometimes APC mutations can be beneficial because they can increase regulation of cell growth, which prevents a lot of tumors from forming in the first place, or sometimes it can lose its whole original function and it can often lead to tumors growing without any regulation at all. So a log-rank test that I conducted on the Kaplan-Meier curve that you can see at the bottom right resulted in a p-value of 0.002, indicating that this was significant. My second gene was KRAS, and KRAS is an oncogene which is used in signal transduction to initiate growth factors. So basically, it's an initiation gene, and it kind of um, basically provides directions for other growth factors to move down a chain, and this allows for a lot of cells to continue their growth. Mutations can lead to the constant activation of MAP kinase, which is an enzyme, and this can cause increased cell proliferation. So cell growth occurs without imminent cell death, and this can lead to tumor growth pretty often. So KRAS mutations occurred in 35 to 45% of CRC cases, which is pretty common, but it often occurred alongside a lot of other gene mutations, so most of them are correlated to each other. The average survival time for patients with KRAS was 43.96 months, and it was 33.97 months for patients with a mutation. So this indicates that the wild type mutation had a higher survival, so a mutation did decrease patient survival. And a log-rank test on the Kaplan-Meier curve resulted in a p-value of 0.0002, indicating this is significant. My third gene was BRAF. BRAF is an oncogene that's also involved in the MAP kinase signaling pathway the same way KRAS is, but BRAF has already been found to have a common prevalence in breast cancer cases, and it has been linked to that. It's also linked to high levels of microsatellite instability, which can determine whether or not cells are prone to overgrowth, and whether or not they're able to regulate their own growth. It also is linked to high rates of preneoplastic lesions. So these are common lesions that are found usually in the large intestine and the colon. And if they are treated early on, they don't often develop into colorectal cancer growth, but without any actual treatment, they can turn into adenomas or tumors. So it does have low mutation prevalence in CRC cases, but when it is prevalent or when it is visible in a CRC case, it is often linked to lower survival. So the average survival time for a wild type patient was 40.44 months, and for a mutated patient, it was 27.49 months, indicating that survival was severely decreased. And a log rank test resulted in a p-value of less than 0.0001, indicating that this does have a high significance. And my final gene was RNF43. RNF43 is a tumor suppressor gene similar to APC, and it negatively regulates the Wnt signaling pathway. So it allows to basically decrease the signaling pathway and its cell growth after an excess of product. So loss of function mutations in RNF33, which are the most common, can lead to enhanced signaling and aggressive tumor biology. So tumors are often very difficult to be regulated once they do start to form, and it's difficult to treat them. So RNF43 mutations are more complicated than the other three that we spoke about because there's been limited research on this gene specifically. So mutations have been associated with both poor prognosis and improved survival, again, depending on the location, similar to APC. Because the location of the function and how many mutation counts there are in a cell do have a severe impact on the cell's function. So that can often determine whether or not it is a positive or a negative mutation. So the average survival time for RNF43 patients was 39.92 months if they were wild type, or 24.85 if there was a mutation. And a log rank test in this resulted in a p-value of less than 0.0001, indicating that this has high significance. In this database specifically, RNF43 cases were limited. However, they did have a strong correlation to survival, which is indicated in the Kaplan-Meier curve. So for my discussion, I did speak about how a lot of the results agree with previous literature about the effects of genetic mutations, specifically on patient survival, and a lot of these genes can serve as prognostic biomarkers for immunotherapeutic efficacy. Just because targeting specific genes, especially before severe cancer growth, so in earlier stages, that can be really, really important to understand how to treat that specific tumor. Early genetic paneling on patients can also provide indicators for more targeted treatments and drug sensitivity. A lot of the genes that I spoke about earlier are sensitive to certain treatments or are able to basically avoid treatment all and like tumors are basically not targeted at all, depending on the gene, because it's really difficult to be able to form targeted treatments for specific patients because everybody's cancer is different. And that's why knowing drug sensitivity and knowing which tumors are more sensitive is really, really important for a patient's survival. So because these are correlations, regardless of whether they're statistically significant or not, they do require further research and investigation. And that's it. Thank you.
Thanks so much. That was really insightful and really appreciate it. If anybody has questions they would like to add, feel free to put them in the Q&A. I will start us off with a few of my own listening in. So can you tell me a little bit more about the data set that you use? Like what kind of scale are we talking about in terms of number of samples, patients?
So on CBioPortal, I can see like the relative size of a lot of these databases. So I did pick one that was a lot larger just because I am testing gene mutations and I don't actually know which patients have which specific counts. It's a lot trickier to make correlations that way. So I picked one with 1,517 patients in it, which was a large enough scale for me to be able to make correlations and have a decent patient size to compare.
Great. And I know you talked about some of them can be used as biomarkers. Are any of them currently being used in
Um,
that way or not yet?
BRAF is being used as a biomarker because it is linked to a lot of other kinds of cancer, specifically breast cancer. But it is still used as a biomarker for all cancer patients, regardless of which type of syndrome they have. Same with KRAS because it regulates an enzyme pathway, so it's a lot easier to narrow down where it's functioning because they can see increased production of certain kinds of products, depending on whether the function is increased or decreased. So it's a lot easier to target that. With APC, it is a well-known gene that it is correlated to colorectal cancer. However, just because it is tricky to narrow down the signaling pathway and kind of regulate it because it's important for so many other functions, it is investigated as a biomarker for colorectal cancer, but not necessarily for treatment, more for basically early diagnosis. But RNF-43 is relatively new, so it's not really being used for that, but it does have the potential to be used for.
Awesome. Thanks so much.
Mhm.
And then just kind of a question about what you want to do next. Is this kind of work that you're interested in continuing in the future, or what are your next steps with this?
I'm really interested in public health research. Um, I am a senior, so this is coming up pretty recently, but I am planning on majoring in cellular and molecular biology. And then I wanted to get my master's in public health, and I wanted to work mostly on research. I think that is what I am most interested but I think I chose colorectal cancer specifically, and I think like it was kind of a little bit more of a random topic, but I chose it just because my family does have a high genetic risk, and I think that really intrigued me because that was something that was not really visible to us. Like genetic risks were not something associated with our lifestyle, so I just wanted to explore that a lot further. But I think research is definitely something that I want to take forward.
That's awesome. That's
Mhm.
great. Yeah, and I feel like I'm seeing a lot of news about the increase in colorectal cancer, and yeah, so
Mhm.
really relevant. Thanks so much.
No problem.